Hello everyone. Today we are going to explore something fascinating, how the body handles dietary lipids. Every time we eat food, whether it's a typical Indian diet with about 20 to 30 grams of lipids per day or a Western diet with 2 to 3 times more lipids, our body performs an amazing process to digest and absorb these lipids. The question is, how does this happen? Let's break it down step by step, starting from the mouth all the way to absorption into the bloodstream. First, let's understand what lipids we actually consume in the diet. The major dietary lipid is triacylglycerol, also called TAG. This makes up the bulk of fat in our diet. Along with this, we also consume minor amount of cholesterol and phospholipids. Now let's look at the structure of triacylglycerol. It's quite simple actually. Triacylglycerol contains a glycerol backbone and 3 fatty acids attached to it. This vertical box represents the glycerol molecule, which has 3 carbons. We number these carbons from top to bottom. Carbon 1, 2 and 3. Each carbon has a fatty acid attached, shown by these horizontal boxes. These fatty acids can vary. They can be a long chain fatty acid or LCFA, medium chain fatty acids or MCFA, or short chain fatty acids or SCFA. Now let's talk about cholesterol. Most of the cholesterol in the diet, about 85 to 90%, is in the free form. The remaining 10 to 15% is in the esterified form, meaning it's bound with the fatty acid. This oval represents cholesterol and this hydroxyl group is very important. So this is a free cholesterol. When this hydroxyl group is replaced by a fatty acid, we get cholesterol ester. Lastly, phospholipids. Their structure is similar to triacylglycerol but with a key difference. They have a glycerol, but fatty acids are only on carbon 1 and 2. On carbon 3, there is a phosphate group with a base attached. Now that we understand what we are eating, let's see what happens when the food enters the mouth. On the dorsal surface of the tongue, there is a specialized gland called Abner's gland. This gland secretes an enzyme called lingual lipase. Lingual lipase works best under acidic pH conditions, but there are 3 major problems in the mouth that prevents effective lipid digestion. Problem number 1 is that the pH inside the mouth is neutral, but lingual lipase needs acidic pH to work effectively. Problem number 2 is that food remains in the mouth for a very short duration, not enough time for proper digestion. And problem number 3, and this is a unique problem. Lipids are hydrophobic, meaning they hate water. In an aqueous environment, all lipid molecules come together due to hydrophobic interactions and forming a large lipid droplet. The problem is that the enzymes are water soluble, so they exist in the aqueous phase, while lipid exist in the lipid phase. So where do they meet? They meet only at the surface. And with respect to size of this large droplet, its surface area is very little, which is accessible to the enzyme. Because of these 3 factors, digestion in the mouth is not significant. But this doesn't mean lingual lipase is useless. We will see why in a moment. After the mouth, food travels through the esophagus and enters the stomach. Inside the stomach, the pH becomes acidic due to hydrochloric acid. Along with this, there is a vigorous muscle contraction called as a churning action of the stomach. This churning action is very important. It agitates these large lipid droplets and convert it into many smaller lipid droplets. Notice what's happening. The surface area is now greatly increased. Plus, food stays longer in the stomach as compared to mouth. So all 3 problems from the mouth are now solved. This is why lingual lipase is not useless. It can now work effectively in the stomach's acidic environment. But wait, there is more. The fundus of the stomach contains a specialized cells called as a chief cells, which secretes another enzyme, gastric lipase. Gastric lipase also works best under acidic pH. However, it is present in very small quantity. Both lingual lipase and gastric lipase act on triacylglycerol, but not just any triacylglycerol. Specifically, those containing short chain or medium chain fatty acids, meaning fatty acids with 12 or less carbons. So let's draw such triacylglycerol structure. This is glycerol backbone and 3 short chain or medium chain fatty acids attached to it. Now these enzymes are very specific. They only break the bond at carbon number 3. This is hydrolytic reaction, so water is added and the fatty acid at position 3 is released. The result is a diacylglycerol, that is a glycerol with a fatty acid still attached at position 1 and 2, but position 3 now has just a hydroxyl group. So this is the structure of 1,2-diacylglycerol. Remember that by this process in stomach, only approximately less than 10% of triacylglycerol is digested, whereas cholesterol and phospholipids are not at all digested in the stomach. Also remember that short and medium chain triacylglycerols are predominantly found in milk and milk products. This is why these enzymes are more significant in newborns and infants who are mostly fed milk rather than in adults. After the stomach, food enters the small intestine. This food is acidic and contains partially digested food called as acidic chyme. This acidic nature stimulates endocrine cells in the small intestine to release a hormone called as a secretin. Secretin has an important job. It stimulates the pancreas to release bicarbonate. What does bicarbonate do? It neutralizes the acidic chyme, making the environment alkaline, which is essential for further digestion in small intestine because lipid digestive enzymes that secreted in the small intestine works best in alkaline medium. Now, the partially digested food in the chyme also stimulates the endocrine cells in small intestine, but this time a different hormone is released, that is cholecystokinin or CCK. Cholecystokinin has several important actions. First, it stimulates the gallbladder to release bile into the small intestine. Bile contains 3 very important compounds: bile salts, phospholipids, and free cholesterol. Second, cholecystokinin stimulates the exocrine cells of the pancreas to release digestive enzymes into the intestine through the pancreatic duct. These digestive enzymes can digest carbohydrates, proteins, and lipids. Since we are focusing on lipid digestion, let me enlist the important lipid digestive enzyme. These are pancreatic lipase, procolipase, monoacylglycerol isomerase, cholesterol esterase, and prophospholipase A2. So, we have 3 things entering the small intestine: bicarbonate, bile, and digestive enzymes. All 3 are crucial for lipid digestion. Let's understand the role of bile. The most important component of bile is bile salts. Bile salts are amphipathic molecules. What does amphipathic mean? It means the molecule has one part that is hydrophilic, that is water loving, and another part that is hydrophobic or lipophilic, that means fat loving, both in a single molecule. I will represent such amphipathic molecule by this schematic diagram, in which this circle represents hydrophilic portion, whereas this tail represents hydrophobic or lipophilic part. When bile salt reaches a critical concentration, they form a special structure called a micelle. In a micelle, multiple amphipathic molecules aggregate in a specific way. Their hydrophilic head points outward towards the water, while their hydrophobic tails point inward, creating a hydrophobic interior and hydrophilic exterior. Now, how does this help lipid digestion? Remember, we had a large lipid droplets, right? The intestinal muscle movement can temporarily break this into smaller droplet, but they quickly reassociate back into larger droplets. But when micelles are present, their hydrophobic interior can trap these small lipid particles inside. Let's show this in the diagram. And similarly for these other small droplets also. So the large fat droplet is permanently converted into many small droplets, each surrounded by or inside a micelle. This significantly increases the surface area of lipid particles. Remember, digestive enzymes are water soluble and can only access the surface. More surface area means much more efficient digestion. This entire process is called emulsification. Now let's see how digestive enzymes work, starting with the most important one, pancreatic lipase. This enzyme acts on long chain fatty acid containing triacylglycerols. So let's draw such triacylglycerol structure. This is glycerol backbone and 3 long chain fatty acids attached to it. But there is a problem. Pancreatic lipase is actually inhibited by bile salt. This seems contradictory, right? We need bile salt for emulsification, but they inhibit this enzyme. This problem is solved by colipase. Initially, we have pro-colipase, which is inactive. It gets activated by the protein digesting enzyme, that is trypsin, to form active colipase. This active colipase forms 1:1 complex with pancreatic lipase, 1 colipase for each pancreatic lipase molecule. Then colipase also binds at lipid-water interface. By doing this, colipase does 2 brilliant things. First, by binding with pancreatic lipase, colipase removes inhibition of bile salt on pancreatic lipase, so enzyme can work uninhibited. And second, since colipase itself is bound to lipid-water interface, it keeps pancreatic lipase exactly where it's required, and that is at the surface of lipid droplets. So, you can say that colipase anchors pancreatic lipase to the surface of lipid droplets. Now, pancreatic lipase can work effectively. Its favorite bond to break is at carbon 3. It does this very rapidly. It can also break the bond at carbon 1, but much more slowly. First, it removes the fatty acid from carbon 3, then eventually from carbon 1. So, 2 long chain fatty acids are removed. The result is glycerol with a fatty acid only at position 2 and hydroxyl group at position 1 and 3. So this is the structure of 2-monoacyl glycerol or 2-MAG. What happens to this 2-monoacyl glycerol? There is an enzyme called monoacyl glycerol isomerase that performs the isomerization. This enzyme transfers the fatty acids from carbon 2 to carbon 1, producing 1-monoacyl glycerol or 1-MAG. That is glycerol with a single fatty acid at position 1 and on position 2 and 3 with hydroxyl group. However, and this is important, 2-monoacyl glycerol is a very poor substrate for this isomerase enzyme. The conversion happens very poorly. So very little 2-monoacyl glycerol gets converted to 1-monoacyl glycerol. This 1-monoacyl glycerol can be further digested by pancreatic lipase. Remember, pancreatic lipase works slowly on carbon 1, but it can still remove this fatty acid, giving us a glycerol plus long chain fatty acid. Here is the key point. Because the isomerase works poorly and pancreatic lipase is slow on carbon 1, triacylglycerol is not completely digested. Most of it remains partially digested. In an average adult human, after digestion, approximately 72%, that is about 3/4, of triacylglycerol is in the form of 2-monoacyl glycerol, approximately 6% remains as a 1-monoacyl glycerol, and only around 22% is completely digested to glycerol and long-chain fatty acids. Let's move to the cholesterol ester digestion. Schematically, it can be represented like this. This is cholesterol and this is its attached fatty acid. Remember, cholesterol ester cannot be digested in the mouth or stomach. It can only be digested in the small intestine. The enzyme that works here is cholesterol esterase, also called cholesterol ester hydrolase. It's a pancreatic enzyme and bile salt is required for its action. This is a hydrolytic enzyme, so water is added and it breaks the bond between cholesterol and fatty acid. The products are free cholesterol and a free fatty acid. Remember, free cholesterol that's already there in the diet doesn't need digestion because it's already free. Now for phospholipids. Let's first draw schematics of phospholipid. So this is a glycerol with fatty acids only on carbon 1 and 2. On carbon 3, there is a phosphate group with a base attached. The enzyme here is a pro-phospholipase A2 which is inactive. It gets activated by trypsin to form active phospholipase A2, and here also bile salt is required for its action. Phospholipase A2 specifically breaks the bond at carbon 2 of the phospholipid. Water is added, the bond breaks and fatty acid is released. The remaining compound has a glycerol with a fatty acid at carbon 1, a hydroxyl group at carbon 2 and the phosphate base group still at carbon 3. This is called lysophospholipid. Let's summarize what we got as an end product of lipid digestion. From triacylglycerol, we get glycerol, fatty acids. Fatty acids can be short chain, medium chain and long chain, and 2 monoacylglycerol and 1 monoacylglycerol. From cholesterol ester, we get free cholesterol and fatty acids. From phospholipids, we get lysophospholipids and fatty acids. Now, here is something very interesting. Most of these products are more or less amphipathic. They have both hydrophilic and hydrophobic part. Exceptions are that glycerol is more hydrophilic, whereas most long-chain fatty acids are hydrophobic. Earlier, we had seen that bile salts are amphipathic, so it can make micelles like this, where hydrophilic head is on the surface, whereas hydrophobic tail remains buried in the center like this. Now, in addition to bile salts, we have many different amphipathic molecules which can also participate in this micelle formation. Suppose this is fatty acid, this is 2 and 1 monoacylglycerol, this free cholesterol, this one lysophospholipid and long chain fatty acids in the center as they are hydrophobic. So here, as you can see, along with bile salts, all these lipid digestion end products are also there in the micelle. Since there are multiple types of molecules in this micelle, we can call it as a mixed micelle. These mixed micelles are still in the intestinal lumen and specifically in the jejunum. Suppose this is the intestinal mucosal cells. This is its apical surface which had a water layer on it, and this is the basolateral side. On this side, cell is in contact with capillaries which drain into portal vein and lymph lacteals. Now here, micelle surface consists of hydrophilic parts while the center contains hydrophobic part. This arrangement allows them to interact with the unstirred water layer on the surface of intestinal cells in the jejunum. This allows diffusion of all digested lipid products into the jejunal cell. But remember that bile salts do not get absorbed in the jejunum. They move further down the intestine and then they are reabsorbed in the ileum. And one more thing, glycerol being hydrophilic can diffuse through the apical cell membrane. It doesn't need micelle for its absorption. So inside the jejunal mucosal cells, we now have glycerol, fatty acids of all types, short chain, medium chain, long chain, 2 and 1 monoacylglycerol, cholesterol and lysophospholipids. Here is where things diverge. Glycerol and short and medium chain fatty acids can directly diffuse through the basolateral membrane and enter directly into the portal blood, which goes to the liver and then to the systemic circulation. However, long chain fatty acids, monoacylglycerols, cholesterol and lysophospholipids take a different route. They enter the smooth endoplasmic reticulum for further processing. Suppose this square represents the smooth endoplasmic reticulum, so all these end products enter into this. Inside the smooth endoplasmic reticulum, some magic happens, which we will talk in a while, and by this magic, these molecules along with some protein are assembled into a large structure called as a nascent chylomicron. Chylomicrons are very large. So large that they cannot enter the blood capillaries. Instead, they exit the cell by exocytosis and enter the lymph lacteals. From the lacteals, they travel through the lymphatic system, collect in the thoracic duct, and finally enter the systemic circulation. Now, the final question about magic in the smooth endoplasmic reticulum. What exactly happens inside the smooth endoplasmic reticulum so that digestion products are assembled in a nascent chylomicron? Inside the smooth endoplasmic reticulum, these broken down forms of lipid are reconverted to their complex form. Let's see how. First, long chain fatty acids are activated. The enzyme fatty acyl coenzymase synthetase, also called thiokinase, converts a long chain fatty acids to acyl coenzyme A, which is the activated form. This requires a coenzyme A and is energy demanding. It uses two high energy phosphate bonds from ATP, converting ATP to AMP plus pyrophosphate. This activated acyl coenzyme A can now donate fatty acids to other molecule. This both 2 monoacyl glycerol and 1 monoacyl glycerol needs 2 more fatty acids to become a triacyl glycerol again. These required fatty acids are donated by acyl coenzyme A and coenzyme A gets released. But here is an important point. They use completely different pathways. 2 monoacyl glycerol is converted through the monoacyl glycerol pathway, while 1 monoacyl glycerol uses the phosphatidic acid pathway. Triacyl glycerol is very hydrophobic, remember this. Next, cholesterol is reconverted to cholesterol ester by receiving a fatty acid from acyl coenzyme A with release of coenzyme A. And similarly, lysophospholipid receives a fatty acid and becomes the phospholipid again. Cholesterol ester is also very hydrophobic, while phospholipid is amphipathic. So notice the pattern. In the intestinal lumen, complex lipids were broken down to simpler forms for absorption. Once they are absorbed and inside the intestinal cells, they are reassembled back to their original complex form. So the breakdown was just to get them into the cell. Now, how do these reassemble lipids from chylomicron? The intestinal cells produce a very important protein called apolipoprotein B48 or ApoB48. When this protein is present, triacylglycerol, cholesterol ester, phospholipids and ApoB48 comes together and form an assembly. This assembly is arranged as a spherical particle. This arrangement is very specific. In the center, we have hydrophobic molecules, triacylglycerol and cholesterol ester, because they hate water. On the surface, we have molecules with hydrophilic parts facing outwards, that's ApoB48 and phospholipids. And obviously, as triacylglycerol are major dietary lipids, this contains far more triacylglycerol than cholesterol ester. This particle is called nascent chylomicron. Nascent means newly formed. It's not yet mature chylomicron. So by this magic in smooth endoplasmic reticulum, nascent chylomicrons is formed. And that's the complete story of lipid digestion and absorption. Let's quickly recap the journey. Dietary lipids, mainly triacylglycerols, undergo a minimal digestion in the mouth and stomach. The real action happens in the small intestine, where bile emulsifies fat and pancreatic enzymes break them down. The products form mixed micelles and are absorbed into the jejunal cells. Inside these cells, lipids are reassembled and packaged into chylomicrons, which travels through the lymph to reach the bloodstream. This elegant process ensures that even though lipids are hydrophobic and enzymes are water soluble, the body can effectively digest and absorb dietary fats. If you have any question or need clarification on any concept, please write them in the comment section below. If this video helped you understand lipid digestion, please like and subscribe for more biochemistry content. Thank you.